Hello, everyone, and welcome to today's webinar entitled Diving Deep into the Microbiome, How Metabolomic and Microbiomic Approaches Can Elucidate Microbiome-Host Interactions. My name is Heloise. I am a Senior Product Manager at DNA Genotek, and it's my pleasure to be your moderator for today's webinar. We have two fantastic speakers for you today, so let me introduce them to you. First up, we'll be hearing from Dr. Jason Bush, who is the Chief Scientific Officer at Solnul. I've actually been following Jason's work for quite a while now, as he's been researching all things microbiome, so I'm very happy to have him speaking. As part of his role as Chief Scientific Officer at Solnul, Jason has completed 2 clinical trials which have investigated how resistant starch is used by the gut microbiome to influence human health. He has a keen interest in complex biological systems, which has really fueled his desire to understand exactly how Solnul can be positively used to impact our health within the gut and beyond. Jason's been integral in protecting the intellectual property of the Solnul technology and has filed numerous patent applications around Solnul's beyond the gut discoveries, which I look forward to maybe asking a bit more about. And after Jason, we'll be hearing from Ken Chalcraft, who is a senior scientist for metabolomics at DNA Genotek. Ken is an analytical chemist with over 15 years experience in high-resolution mass spec. He specializes in small molecule discovery, quantitative analysis, and general metabolomics research. Ken holds a PhD from McMaster University, and prior to joining DNA Genotek, he was active in clinical toxicology, as well as tobacco and cannabis-based research, focusing on small molecules and metabolite analysis in a diverse range of mammalian and plant-derived matrices. So we're very fortunate to have these 2 great speakers with us today. I hope that you will join in the Q&A after they have both presented, but without further ado, I'll hand it over to Jason.
Thanks again for the introduction. So yeah, I'm going to talk a little bit today about some of the work we've done on Solnul, relating to the metabolomics analysis and kind of how it integrates into our work in the dietary supplement industry. So, Solnul is a type of resistant starch. Maybe it starts best with a definition of what resistant starch is. And so it's a sum of starch and starch digestion products that on average reach the human large intestine. But maybe that's not particularly helpful. It's important to know that starch has kind of been an important part of human diets since basically before we were humans, all along our ancestral history. And kind of further to that, starch is really the only complex carbohydrate that our body has the enzymes able to digest. And so when we consume starch, generally its digestion begins in the mouth, ends in the small intestine, and little parts are carved off, and starch is made up of glucose. Glucose, of course, is an important fuel for our body. But as this definition indicates, our ability to kind of convert starch to glucose is incomplete. And the remainder of that forms an important subtype of dietary fiber called resistant starch. Now, as I mentioned, historically, we've kind of consumed starch for a long time. In the Neolithic period, so that would have been kind of early farming era, estimates are that we were getting about 50 grams of resistant starch per day. And these high levels persist in certain developing nations. And because resistant starch, for reasons I'll explain in a second, is actually hard to get from the diet, it's kind of spurred a subset of research and why it's important for human health. And so we've been able to identify kind of like minimum thresholds necessary for things like bowel health and metabolic health. So if resistant starch is important to our diet and it's a part of eating a starchy diet, and we still eat a starchy diet, is there even a problem here? Well, the answer is yes. And this relates to really how modern preferences in food, taste, and texture, as well as our ability to prepare and especially to store starchy foods, has dramatically decreased the amount of resistant starch we're getting from our diet. Modern estimates are somewhere between 4 and 10 grams per day in developed nations. Now, in our space, the dietary supplement space, we interact a lot with dietitians who suggest that, you don't need dietary supplements. What you should really focus on is getting these nutrients, these important nutrients from your diet. Okay, so if we try to take that approach with resistant starch to get that 15 grams per day for metabolic health, we turn to our number one source of resistant starch in the Western diet, and that's actually French fries. And I think we can all agree that there is no model in which somebody's actually going to consume 10,000 calories of French fries and get that 15 grams of RS and somehow achieve some sort of a metabolic benefit. So there is a deficit of resistant starch consumption in the Western diet. Now, one of the things that's kind of plagued resistant starch research, clinical studies, has been the fact that, yes, now we're interested in the microbiome. We recognize RS as a form of dietary fiber, but historically, RS, the recognition of RS actually predates a lot of that conversation. And the initial studies looking at RS were around glycemic impact. So the thinking was that if you could take a cookie or a muffin or a piece of bread, and you replace the digestible starch in that food item with resistant starch, you can blunt the glycemic impact, right? You can reduce that postprandial glycemic effect. And so initial studies in RS were really based around this fact. And then somewhere along the way, somebody said, hey, you know what? If this isn't digested, it's probably going to be fermented by the microbes in the gut. So let's see if there's an effect there. Now, the odd thing that happened was that when they started doing those microbiome studies, they stuck with these really high doses. And so you can see from this slide here, some of these doses are looking at giving people 30 to sometimes even 70 grams of RS containing product per day. Now, that may work if you're feeding somebody kind of, modified bread or something along those lines. But from a dietary supplement perspective, it's just a non-starter. There's not enough room in that pouch or that container of protein powder or whatever it may be to fit a 30-gram dose of RS in there. So we recognized this opportunity probably about 5 years ago. And so we initiated the world's first resistant starch study. We did this work with Nutrisource, a contract research organization out of Guelph, Ontario, and it was set up as you see here. Now, we looked at two different doses. We kind of positioned our doses around what other prebiotics typically are, somewhere in that 5 gram per day dose range. And so we looked at 7 and 3.5 gram doses. Now, for the purposes of the discussion today and for most of our research moving forward, we're really focusing on that low dose arm, that 3.5 gram dose, for a couple of reasons. One, we really didn't see any differences between the high dose and the low dose. And two, people don't want to add more material than they need to because it just costs them more money. So what did we see? Well, initially, we kind of wanted to look at changes in the microbiome and improvements in host health, because that's really how you meet the criteria for being a prebiotic. And so we saw significant increases in bifidobacterium, which is not surprising. This is a primary degrader of resistant starch. But we also saw increases in Akkermansia. And that was kind of surprising. I won't talk too much about it here, but we have some thoughts as to what's going on there. So that was the microbiome component. And then in terms of the human health component, we also studied stool form. And so what was interesting here is that we saw improvements, in other words, reductions in both constipation and diarrhea-associated stool scores. So from that prebiotic definition, we're improving the microbiome and we're providing a host health benefit, and that was great. But we were optimistic. We thought if resistant starch is really as important as everybody says it is, it's an important part of the diet that's now been stripped out, we think that there may be more opportunities, more health benefits than just looking at stool changes. Now, I think we can all appreciate, especially on this call, that there's a lot of interest in the connections between the gut and other parts of the body, whether that be the gut-brain axis, the gut-skin axis, the gut-heart axis. There's something going on. There are connections between those microbes which are trapped within our gut and these other parts of our body. Now, there are different ways in which the microbiome can do this, and we were really focused around looking at blood for a couple reasons. One, we know that certain microbially modified or derived metabolites are present in the blood and circulating. A great example of these are bile acids, right? So these are substances that are produced by the liver, released by the gallbladder, and they help us to absorb fatty foods, but they're extensively modified by the microbiome, such that most of the bile acids circulating in your blood are derived actually from microbial activities. And they make a great candidate for this signaling molecule. And then the flip side is that we're also able to capture information about host health responses. And that's what I'm going to start my talk on, or my kind of research focus here, is really around histamine. So what is histamine? Well, it's a mediator, a local mediator of inflammation. A lot of us are familiar with this. People take antihistamines to reduce the symptoms associated with seasonal allergies. We saw with Solnol at that 3.5 gram dose a significant reduction and a clinically meaningful reduction in histamine levels. And so this was great, but how did we get here? How could this possibly be happening? And one of the things that I like about doing metabolomics research is that it really pushes my knowledge as a scientist. I had never really thought about histamine before this. One of the things in reading up on histamine is the recognition that most of the histamine in our body is actually trapped within a gut. Okay. And that's for a couple reasons. One, it's in foods we eat, and two, it's produced by a subset of bacteria. And so the obvious question is Solnol influencing the abundance of these histamine-producing bacteria? And the answer is kind of, maybe. We saw significant reductions in 2 histamine-producing bacteria. And because these weren't particularly abundant bacteria, they're not particularly histamine-heavy producers, we thought, well, there's probably more to this going on. So, and this kind of highlights the beauty of doing metabolomics work, is that you can do kind of pathway analysis. So histamine, for example, is degraded by 2 main enzymes in the body, diamine oxidase and histamine N-methyltransferase. And kind of hinted at this on this slide here, but you'll see, so the hypothesis is that if we see reductions in histamine, and Solnol is achieving that by enhancing conversion to methylhistamine, we'd see a concomitant increase in methylhistamine. We don't see that. If anything, we see an increase in the placebo group. So there's no effect there. Now, the major degrader of histamine is diamine oxidase. Unfortunately, we didn't capture the breakdown product of diamine oxidase, but that's fine because diamine oxidase also degrades other metabolites, including polyamines. So again, if Solnol is degrading histamine via diamine oxidase, we'd expect to see a similar pattern in these polyamines, but we don't see that. So are we at a dead end? Well, not really. Again, kind of in the process of learning about these different metabolites, what their biological role is, I came to realize that polyamines are dynamically regulated at the cellular level through acetylation. So acetylation kind of marks them to be kicked out of the cell and to be degraded. And there's a direct connection between acetylated polyamines and intestinal function, in that there's a correlation, a positive correlation between acetylated polyamine levels and lesion severity in inflammatory bowel disease. And so this got me thinking, if histamine, the main reservoir of it in the body, is actually in the gut, we're seeing a reduction in histamine when people take Solnol. Could it be that Solnol is actually improving intestinal barrier function to keep histamine in the gut? And so if that were happening, we'd expect to see a reduction in these acetylated polyamines. And that's something that we do see. Okay, so this is exciting. This is kind of directional. Now going into the literature, what other groups of metabolites can we look at to potentially test out this hypothesis of enhanced barrier function further? Well, there are a couple of studies showing that a subset of amino acids are characteristically elevated in individuals with leaky gut. And again, we see significant reductions in those amino acids. Importantly, maybe the most important one here is 5-hydroxylysine, because this is a marker of collagen degradation. A lot of us will be familiar with collagen in the context of skin health, these sorts of things. But remember that the organization of the skin is very similar to the organization of the intestinal epithelium, and that collagen also plays an important role in that basement layer. So what we're inferring from this is if we have reductions in circulating levels of collagen breakdown products, we're likely helping to support that collagen barrier. And then the third category of metabolites that we were interested in looking at are carnitines. Now, carnitines are important mediators. They shuttle fatty acids into the mitochondria for beta oxidation. And it's been shown previously that when you have dysfunction of the mitochondria, this oxidation doesn't go to completion, and you end up with an accumulation of short and medium chain carnitines. Now, how does that relate to intestinal barrier function? What's been previously shown in a couple of different studies, but especially in terms of non-steroidal anti-inflammatory drugs, that these have an adverse effect on mitochondrial function, and then that results in impaired tight junction function between those enterocytes. And so in other words, when we're looking at these data, what we see is a significant reduction in those incomplete breakdown products. So the inference here is that we're seeing improvements in mitochondria function. And if we have improvements in mitochondria function, we should also have improvements in intestinal barrier function. So kind of stacking up these different aspects of metabolomics, we're seeing a reduction in serum histamine levels. We don't see any meaningful effect in terms of microbiome changes. We're not seeing an effect on the enzymes that degrade histamine, but we are seeing improvements in acetylated polyamines, which, as I mentioned, are a marker of lesion severity inflammatory bowel disease. We're seeing reductions in kind of a fingerprint of amino acids that have previously been characterized to be elevated in those individuals with leaky gut. And finally, we're seeing improvements in mitochondrial function. So helping to improve that tight barrier function, tight junction function. Okay, so the final thing we did was to look at correlations between changes in these markers of leaky gut and changes in some of the bacteria that we had previously documented as being elevated. Because I'll talk about this more in a second, there's kind of this expectation in the prebiotic definition that if you're stimulating the growth of certain bacteria, that the changes in those bacteria should actually be correlated with the host health improvements. And while we saw a little bit of a connection with Akkermansia, we saw no significance in a connection between Bifidobacterium and these markers. And so what I've got here, this is the ISAP definition of a prebiotic, stating that it's a substrate that's selectively utilized by the host microorganisms, conferring a health benefit. And in the bottom, I'm just kind of showing how characteristically prebiotic studies have been set up, where you have consumption of a prebiotic, it gets fermented, people look at changes in the microbiome, they hopefully see increases in beneficial microbes, and then they're also measuring a health benefit. Remember, in our case, we initially looked at stool scores and improvements there. But when you look at the prebiotic clinical trial literature, you really don't see a lot of correlations between the changes in those beneficial microbes and the changes in the health benefits. And so what we're thinking now is that we're kind of missing part of the story. And we're changing our thinking, and we've recently initiated a study to look at stool metabolites because we feel that it may not be so much changes in the beneficial microbes that are correlated with host health benefits, but changes in those metabolites that are locally produced. And so that's a story that we're hopefully going to be able to share sometime in 2025. The samples are being run as we speak. And so I think that kind of sets up Ken's talk a little bit here as well. So before I end, I just want to thank our collaborators, as well as acknowledge the Ag Action Canadian Agricultural Partnership for funding that helped to support this work. So with that, I'll throw it to you, Ken.
Okay, today for my presentation, I'm going to be focusing on what are some of the more unique aspects to performing a metabolomic study using stool as a representation of gut activity. Because whether you're new to the field of metabolomics or you're a veteran that's primarily been looking at more classical sample types like blood and urine, there are a number of peculiarities and challenges with gut sampling which should be taken into account before you begin your study in order to get the best possible results. Now, a good way to think about the gut is as an independent ecosystem where a huge diversity of microbiota reside. And a good representation of this ecosystem is found in the stool, where the bulk is primarily made up of bacterial biomass. For each of the bacteria of species that are present, each has its own biochemical machinery and prefer ways to metabolize or break down nutrients. And some bacteria prefer to use metabolic pathways which produce metabolites that have a beneficial impact to the host, while others prefer pathways which produce metabolites that have a negative impact. Beyond their individual metabolism, each bacteria can have a profound effect on its neighbors and the overall ecosystem. These impacts can range from active inhibition of its competitors through the production of metabolites, or it can overall have an impact to its environment due to changes in pH due to the acidic or basic nature of the metabolites that are produced. And of course, what we feed them in our diets can also have a massive impact on what the bacteria are actually doing inside of the gut. Now, in general terms, we often refer to gut bacteria as either being good or bad for health. And often what differentiates them is how they actually metabolize nutrients and how they impact their neighbors and the overall gut environment. Now, as I mentioned before, there are some species which prefer to produce metabolites which are known to have beneficial or deleterious effects on the body. And there are numerous examples in the literature looking at how metabolic outputs from various bacteria can in turn lead to some pretty profound implications for human health. And furthermore, there are a surprising number of metabolites, as Jason mentioned, which they can be found in blood, but they can only be really explained due to gut bacteria metabolism. We simply just don't have the machinery within our own bodies to produce these molecules within our own cellular biochemistry. And the overall value of metabolomics in terms of a multi-omics approach to gut research is that by knowing and understanding how these bacteria are actually responding metabolically in the gut to various conditions, such as diet and just the mere presence of other bacteria, it can lead to better health outcomes via treatments such as probiotics or dietary supplements, in addition to more classical pharmaceutical approaches. Now, unfortunately, measuring the metabolome in stool is very rarely easy, and one of the big reasons why is the difficulty in sample collection and the overall instability of stool. And I'll talk about each of these points in a little more detail shortly, but just some of the challenges that come with doing gut metabolism is rapid secondary metabolism and fermentation, which happens outside of the gut after defecation, the difficulty in quenching secondary metabolism, reproducible sampling within the stool, and the fact that most people really can't give a sample on command. Typically, they have to wait till nature calls, and that's usually not in the best time for the lab to actually collect the sample. And as you can guess from the smell of feces, many metabolites are very highly volatile, and they can be lost very rapidly if not trapped. So probably the most difficult challenge in measuring the gut metabolome is the very high activity which is present in stool, since it's primarily made up of independent cells that want to survive outside of the gut just as much as they do inside. And as well, there's a lot of active enzymes which may be present outside of the cells in the stool. One of the major processes, which is a very high interest for gut metabolism, is the production of short-chain fatty acids, which can be especially active outside of the gut environment. And this in turn may lead to other secondary effects just due to the pH changes which happen once you produce more and more of these acids. And because any of these changes which occur outside of the gut do not reflect what's actually happening inside of the host environment, which is really what we care about, it is imperative if you're going to be going down this avenue that you stop the metabolism and any metabolites are stabilized as soon as possible after stool collection. Now, there are typically two ways to stop secondary metabolism in stool. The first involves immediately freezing the stool after collection. And if you can collect the sample in a clinic or other controlled lab setting, this is kind of ideal. However, as mentioned earlier, more often than not, most people can't give a stool sample in a clinical setting. They need to collect it at home. And for at-home collection, trying to use flash freezing, this typically involves the donor needing to place a sample in their freezer in their kitchen right next to their food. And as you can imagine, there's a natural reluctance to do that, even if it is in a sealed container. Most people just don't want to put their poop where they eat. And even if they do kind of follow that, there's still issues within cold chain shipping. So again, if the stool warms up in between collection at home to the lab, it can have a profound impact on the metabolic signature within the stool. And because of this, there's a very high risk of non-compliance with an at-home collection using flash freezing. The other major approach is using either a solvent or chemical stabilization approach, where you add a solvent to the stool after collection. One of the major advantages of this is it allows for room temperature stabilization and storage, typically within at least a week of collection on here. One of the issues with this is if you are going to use a stabilization approach on this, you need to immediately homogenize the stool with an appropriate stool to solvent amount. The container that's being used also should be very airtight to reduce the loss of volatile analysis and as well just prevent leakage within kind of transportation. And finally, the sample device must be compatible with the solvents to reduce leaching of any plastics or rubberizers that might be present within the device. And finally, anything that you're going to be adding to the solvent should be compatible with the final analytical platform, which is usually mass spectrometry. Now, as I mentioned, immediate homogenization, if you are going to be taking a solvent stabilization approach, is important for the simple reason that if you just dump solvent onto a big clump of feces within a container, the only cells that are actually going to be stabilized are on the peripheral of the clump on that. Anything what's in the interior, if it's not exposed to the solvent, is still going to be metabolically active. And as well as I mentioned, if you want to reduce the amount of headspace that's actually happening within your container, because any volatile analytes that are happening, they can very easily leave the solvent and be kind of lost when you open up the container. So to show 4 different scenarios that could happen when you actually collect stool, we did a short demonstration where we took an equal amount of stool placed in a tube with a defined amount of ethanol-based solution. And to cue this up. So the first example I'm going to show is when we have no mechanical means of homogenization present. It's just simple shaking it with some solvent on here. And as you can see, there's not much of the stool that actually gets into the solution. The second is when we add a steel ball bearing, which does help break up the stool a little bit, but again, there's still not much of the stool that gets into the solution. The third example is pressing the stool through an X-shaped aperture, which does break up the stool. However, there's still some pretty major chunks that are happening on here. And the fourth example is when we use a ball bearing plus the press, which gives a nice slurry with minimal clumps at the bottom. So again, most of this, even with additional shaking, is still not getting a lot of the stool within the solution, or there's big chunks, which of course are not going to be stabilized. Another challenge beyond the stabilization is just how variable stool can be in terms of its actual quantity and quality on here. So again, within the Bristol stool chart, there's going to be a huge variation in terms of texture, hardness, and water content that's actually happening on here. And for most people who are collecting at home, it's just not practical for them to try to weigh out a consistent amount of stool to be able to be stabilized. And even in the lab, it's still not easily to be able to weigh out an accurate amount of stool simply because of odor control and really biohazards constraints that needs to be taken into account, and as well as very time consuming. Again, as we saw from the need for homogenization, optimal amount of stool to solvent is needed for stabilization. So internally, we have developed a product that can at least address some of these aspects of sampling. So our OmniMet gut. This was the fourth example within the video. It does have within itself a homogenization mechanism, which can break up the stool quite easily. And it can also fairly reproducibly collect about 500 milligrams of stool. For the most part, this is fully compatible with most mass spec derivatization, most mass spec protocols and derivatization protocols within, typically used within metabolomics. Okay, another issue which does come up quite often with doing metabolomics is the need to use targeted methods for metabolomics. So no matter how good your untargeted method or discovery platforms are, there's always going to be a few metabolite classes that they simply don't give very good response, especially within mass spectrometry. Typical examples of this are short-chain fatty acids, biogenic amines, and as well monosaccharides. For these particular compounds, you may or may not be able to detect it with a non-derivatization approach, but however, because of the sensitivity and usually poor chromatography, a lot of times the data is not very reliable. So you can benefit from using a targeted approach and kind of knowing before you start the analysis whether you're going to need some of these targeted approaches. It's a good idea to kind of work out before you start your analysis, because this is of course going to impact the cost and the amount of time that's required for analysis. Now, one challenge that we kind of learned the hard way and during gut metabolomics is even if you do have a good collection and you do have a good analytical platform, you still may not get the results that you want. So one internal study that we did where we wanted to look at the metabolomics as part of a multi-omics assessment of intermittent fasting in the human body. So for this particular thing, we had donors fast for a particular amount of time, and then we measured the metabolome. We discovered once we started really looking into the data, we did see some changes specifically in histidine, lysine, and tyrosine and secondary bile acid pathways. However, the overall data quality was quite weak. It didn't really give us that big aha moment where it kind of really demonstrated what was happening during fasting conditions. And one of the reasons that that was happening on this is some donors did show a very robust metabolic change. Others didn't really show much of a metabolic change at all. It was really quite muted, and that kind of threw our statistical analysis in terms of looking at the control groups into disarray. Once we started digging a little further into the literature, we discovered that we probably should have taken into account gut transit time. So as you can imagine, once you eat something, it's not immediately that the impact is going to be felt by the bacteria within the gut. It takes some time for the nutrients to get through the stomach, through the digestive tract. And it's been shown in the literature that the gut transit time, how long it takes to actually get through the gut, can have a profound impact on the production of metabolites within the gut. The other thing that should be mentioned is the flow of nutrients and bacteria through the gut. It's not a linear process like through a straight tube. Rather, the gut has a lot of folds and nooks and crannies that things can become trapped in. So things can kind of get spread out within the gut as it's being metabolized. Now, to be able to measure the gut transit time within metabolics experiment, there's typically two ways of doing it. The first is using what's known as the SMART pill, where it can actively collect various ambient conditions such as pH and temperature throughout the digestion process, which does have some advantages. However, it's fairly expensive, and again, it doesn't really take into account the fact that it's a turbulent flow through the gut. The other major way of doing it is using a dye marker, which is commonly used as the blue poo method after the blue dye that was initially used in the first one in 2021, where a donor consumes a blue dye, and then they kind of measure that as a measurement of the gut transit time. So to be able to assess this for our own study, we did a limited pilot study where we had 5 donors consume blue dye E133, and we measured the intensity of the dye through over 4 days where we collected that. And sure enough, we did notice that of 3 of the donors, the dye was passed fairly quickly, although not all at once. For all of the donors, even up to day 2, we were still detecting some of the dye. Whereas some of the other donors, it was much more spread out, with one donor passing almost 25% of the dye as late as day 3. And this, again, kind of leads credence to one of the issues that we probably had within our dietary study is we were simply missing the really big impact of the fasting conditions just because the gut transit time was too slow in some of our donors. Another common issue within metabolomics that comes up, especially within gut metabolism, is you need to choose your sample cohort size very carefully. So this is true of all sample types, but within gut, there's a huge variability, even within healthy donors, of your typical range of metabolites. So for an example, deoxycholic acid, which is a very common bile acid, it's got a huge natural range, even within healthy samples. So what can happen is if you do not have an adequate sample cohort, a lot of times you can just miss what you're actually looking for simply because there's just too much variability. Now, within the literature, there are sample size calculators available. The 2 primarily ones that I've used is MetSizer and S-Sizer. However, even if you don't want to use that, I would highly recommend just use as high a sample cohort as practical with a recommended minimum of an N of 7. Another challenge that comes up, especially within gut metabolism, is it's very, very difficult to get an accurate assessment of your data quality. Again, for the most part, you can analyze any biological material. It'll give you a metabolomics output on that. It doesn't necessarily mean that it's actually a good quality data set reflective of what you're trying to study. And within gut metabolomics, it's especially challenging because there's not a standard reference material that does exist right now for stool analysis, although NIST is working on one. For those of you that are not really familiar with the use of standard reference materials, these are samples that are very well characterized, and you can analyze them along with your other sample cohorts to be able to demonstrate that your analytical platforms and extraction profiles actually did work correctly, and it can be used to demonstrate overall quality. Because an SRM is really not available with stool samples, usually what's done is you use a pooled sample where you take a little aliquot of everything and use this in lieu of a standard reference material as a quality assessment. Other standard reference materials can also be used, such as plasma, which is available through NIST to be able to complement your pool quality control results. However, if you are new to the field of metabolomics, I highly recommend that you check out the Metabolomics Quality Assurance Quality Control Consortium. This is a group of leading metabolomics researchers that have put together a number of articles and recommendations in terms of good working practices, which will help guide your metabolomics assessment before you start. This way, it'll help you gain a really good assessment of whether your metabolomics data really is valid, and it gives a lot more credence to your analysis, which will help you publish a little later on when you do have good data. And finally, the last challenge that I'm going to bring up, which is both the challenge and exciting, is we are just scratching the surface of what's actually going on in terms of a metabolic output within the gut. So a very good example of this, which was recently published, where a researcher or researchers looked at a retrospective analysis, which they called reverse metabolomics, and they discovered that there's a huge plethora of new bile acid conjugates that, for the most part, they've always gone unreported because there's simply no good standards out there to help on the identification. So there's probably a lot of metabolites that are being produced within the gut that we simply don't know anything about, and we don't have a standard to be able to really get a good identification for it. And within the kind of overall multi-omics approach, it can be a huge help to let your metagenomic and metatranscriptomic data kind of help elucidate what metabolites might be present. So for instance, if you know a particular bacteria is present, it may help you guide kind of which metabolites might be really impacting your health analysis. And with that, I'll open it up for general discussions and questions. Thank you.
We have a couple of questions that are around the same theme, so I'm going to go with it and we'll see where it takes us in terms of discussion. Jason, did you, in what you presented, did you do any work to connect the metabolomics profiles to any specific microbes? Is that in the works?
Yeah, that's a great question. So with the work that we've done, the work that I presented here today, it's all from humans, right? So there's no opportunity to kind of do any sort of mechanism of action. We're really relying on correlations at best to kind of imply or to understand those connections. I think the question, can we use stable isotopes to measure fermentation profiles and kind of the generation of metabolites? It's a great idea, certainly one that we're interested in. But for our purpose, we've got to translate whatever our discoveries are ultimately to a commercial application. We're not just interested in publishing papers. So what we've done so far is kind of try to connect some of the more important metabolic changes, metabolomic changes that we're discovering, and try to find ways to take those into an in vitro system where we can do those types of experiments.
Very cool. And yeah, you touched on it, Ken, at the end about potentially using metatranscriptomics to make that connection. Do you want to talk a little bit more about how you foresee that happening? Sorry, on how we could potentially leverage metatranscriptomics to make that connection between the metabolites that we're seeing and which microorganisms are involved in the process.
Yeah, I can take that. Again, if you know which bacteria are on it, some of the bacteria that we're in the gut, they are fairly well characterized in terms of their preferred metabolic pathways. If you kind of have a cheat sheet and you know exactly which bacteria are present, it may help you elucidate kind of whether your metabolomics data really makes sense or if it's kind of a false discovery. So kind of using some of that other output might help guide your metabolomics and vice versa.
Very true. I did have one question. Sorry, they're coming in, so I'm trying to keep on top. So what is some next steps, maybe, Jason, now that you guys have had some of this data, what have you learned and where are you going to go next in terms of your research?
Yeah, so it's certainly been directional. So a lot of the changes that we see, so I guess just to kind of back up a couple steps, working in the dietary supplement space, it's important that we do our studies on healthy people. And that's kind of counterintuitive because healthy people don't have problems for the most part. You don't expect, for example, to see dramatic improvements in blood glucose control in a healthy population, because if you drop blood glucose levels, if you're trying to study something that you think might help diabetics and you expect a decrease in blood glucose and you see a 2 millimolar decrease in a healthy population, you're putting those people into a hypoglycemic scenario. It's not healthy. So with our data coming from that healthy population, we're seeing kind of subtle changes, but those that are clinically meaningful and statistically significant. So it puts us in a great position now to take some of that data around histamine. So do we set up a study, a seasonal allergy study? Could it be that consuming Solnol in the springtime with hay fever, things like that, we see a substantial improvement in that sort of symptomatology? Similarly, I touched on 5-hydroxylysine. It's a marker of collagen degradation. So could it be that when you administer Solnol over a long period of time, maybe you see improvements in skin quality, whether it be hydration, reductions in fine lines, those sorts of things. So just long story short, I think the metabolomics has really been, that data set's been a treasure chest for us. And now we're able to kind of use that information to de-risk future clinical trials that are more targeted at those health outcomes.
Thank you. This is a kind of a question for probably both of you, but how do you feel about, or maybe just if you can comment more on your thoughts, Jason, you mentioned you were doing host metabolomics, but there's also that opportunity to do gut metabolomics. How do you feel that or rather if you want to comment on the different opportunities that either or option gives you in terms of the research and really highlight any differences?
Maybe I'll just jump on this first, Ken, and then
Sure.
I'll throw it to you. So our interest in stool metabolomics was secondary. So we felt that the serum metabolomic changes would give us, maybe a look at some of those improvements or some of that chemical messenger signaling coming from the gut. But we really wanted to see exhaustively how many different host health attributes can we improve? What kind of pathways are we improving? And so now we're looking to stool metabolomics as a way to complement that. As I mentioned kind of at the end of my presentation, the ISAP definition stipulates that a prebiotic is a substrate that's selectively utilized by beneficial bacteria, conferring a host health benefit. Most prebiotic studies, most clinical studies can't show a correlation between changes in the microbiome and changes in host health. So what we're hoping is that maybe if we turn over the stone of the stool metabolites, maybe that'll give us a better connection to some of these host health benefits.
Yeah, to add on to that, again, to really think about it, the gut metabolome is really what's available to the host. It doesn't necessarily mean it's going to get into the bloodstream on some of these things. So it's kind of what's really available. And as well, by looking at what's happening in the gut, it does impact certain bacteria, can actually impact other bacteria to the point where they would normally be producing particular metabolites, which may have good or bad impacts to the host. However, just by the virtue of the fact that something else is present, it may be inhibiting that. That's where kind of the multi-omics approach really does come in handy.
Great, thank you both. Ken, do you want to comment a little bit about any quantitative work that can be done on samples and some challenges potentially there?
Yeah, so as an analytical chemist, quantitative is obviously preferred. So one of the issues that does come up with a lot of discovery bases is they're semi-quantitative, which you're really looking at relative differences between multiple groups, a control group versus a treated, let's say. Unfortunately, mass spectrometers, as powerful as they are, they do have some limitations in terms of how reliable the quantitation can be. Again, this is where the quality control really comes in handy, and using some more targeted approaches can be very beneficial, even though it is a little bit more limited in terms of what you can do. In terms of correlating it to quantitative, this does usually lead into phenotype analysis, where just because a metabolite is present, it may not be to a threshold where it's either beneficial or kind of deleterious to the host.
Yeah, thanks. Just a quick one. You mentioned the blue dye work. Do you know of any other dyes that could be used for tracking that transit time?
I haven't seen a whole lot in the literature. I think once the blue dye came out, that was one that most people kind of adopted. My guess is there's plenty of other dyes that could work. Internally, we've only used the blue dye, but my guess is there's other food dyes that probably would work just as well.
Great, thank you. Jason, I'm curious because you've clearly highlighted how, to your point, we might be actually missing the picture if we're focusing in on the microbiome, and the metabolomics portion might actually show a lot more. Based on what you're seeing in the market or in the research space that you're in, how common is it for prebiotic or even probiotic companies or research groups to be doing metabolomics nowadays?
Yeah, I think we were one of the first. I think there are a few others that have kind of gone down this pathway, but I mean, we were really fortunate to get some funding to help defray the cost of it, because it does add up. We did our work with the Metabolomics Innovation Center, specifically the Genome BC Protein Center out in Victoria, and it was kind of interesting to find out in conversations later that we were the largest single project. We had the most samples that they've ever handled for a single project. And so it does, when you're doing a clinical trial, multiple time points, looking for multiple aspects, it does really start to add up from a cost perspective. So I think that's been prohibitive, but I think costs are coming down. So I think we're going to start seeing more people moving into this space. I was having a conversation with a colleague just the other day, and he was asking me about, he does clinical trial work, and he was asking, you know, are these things feasible? And we're starting to see now that we can actually, our customers, our partners in the market are using generated from metabolomics analyses to substantiate claims on their products. And I think that's a real exciting threshold to have crossed.
Yeah, that's awesome. With that in mind, obviously, if you're going to go untargeted for that discovery, that's most of the time very expensive compared to maybe a more targeted approach, which we talked about. Ken, maybe any thoughts on other targeted analyses that might be useful in this space other than short chain fatty acids? I know.
Yep. So again, there's a wide diversity of metabolites that might be useful for that metabolism. One particular class which does come up, and Jason kind of touched on, is biogenic amines, things like putrescine and spermidine and things like that. Unfortunately, with these, just due to their chemical properties, you can detect them with most electrospray systems. However, the sensitivity and the overall chromatography tends to be very poor unless they are derivatized. So you do need to take it with a little bit of caution if you're not actually using a derivatization or a targeted method for these. You can get some data. However, it happens very often where the overall data quality and the reproducibility can be quite poor. In that case, if you do know that, say, your biogenic amines are going to be an important, or at least you suspect, it may be worth taking a targeted approach for that.
We're getting some questions on data analysis side of things, and maybe you can both comment from a different angle. Jason, obviously, I believe that metabolomics data would have been pretty novel for you to be analyzing. How did, you know, what were some of the challenges? How did you overcome them? And we'll hop over to Ken after.
Sure. Yeah. So I relied heavily on the folks at the Protein Center to kind of guide that analysis. We were fortunate, you know, as Ken mentioned, we really focused it on, you know, on targeted metabolomics. So having those internal standards there to kind of give us concentration level information was really helpful. And, you know, with metabolomics, and I think maybe Ken can speak to this more, when you're looking at so many different data points, it can become really important to correct for false discoveries. And so I kind of had to to go back in time a little bit and start thinking about statistics and some more, what are the appropriate models and statistical methods to correct for this multiple testing for these samples. And, you know, that really plays a factor in our space, because again, looking at those changes in healthy people, it's not like we're studying a diabetic population and we're expecting to see big differences between a treatment and a control. But still, fortunately, a lot of those really exciting metabolites have held up despite multiple testing. So Ken, I don't know if you want to go further.
You covered a major challenge with the data analysis. There's the old adage, you know, there's lies, damn lies, and then there's statistics. For untargeted approach, you're always going to be subject to statistical analysis. And again, this is where coming to things like having an adequate cohort size, it can really make or break your analysis, to the point where if you're not capturing that kind of natural variability, you can either have many false discoveries, especially if you're looking at thousands of metabolites, or you just may actually miss something because the natural variability is so high that you might have one outlier, which is really important, but you just didn't have the statistical power to be able to really identify it as being something that's worthwhile looking into.
Right. And just jumping off of the same idea of, there's a lot of cost to doing multi-omic studies. Just if you have any thoughts about when it's appropriate to potentially do metabolomics in a microbiome study, or maybe you can speak to your experience, Jason, as to what you've learned from your studies as to when was the right time to run those analyses on the samples. I'm guessing you also have, more studies in the works where you're planning on doing metabolomics, but now that you know a little bit more, how are you curating your studies to integrate these multi-omics?
To be honest, I don't know that we will. I kind of feel like the metabolomics data, as I mentioned, was kind of very directional, and it may be guided and de-risked some future clinical studies for us that are really targeted on finding those patient outcomes. So setting up a clinical trial, the more analyses you add, the more samples you collect, the greater the chance that a participant's going to get frustrated and drop out. And two, the cost really goes up from that contract research organization perspective. So for us, the investment early in metabolomics, I think, helped to guide those future studies in such a way that we may not need to do things as exhaustively. And then kind of conversely, for those stories or those metabolites where we really want to get at the mechanism of action, level of understanding, I think we're going to go back and almost do maybe more of an in vitro approach to test those things out. As that one question mentioned, if we can look at isotope labeled resistant starch, for example, maybe we can trace kind of what's actually happening with that metabolite, what it's feeding into in the microbiome, and what that output is. So I think that's kind of what we're thinking right now. Not to say that we wouldn't do another big study again, but I think if we did that, it would have to be kind of towards a greater good. It wouldn't be such a discovery process.
Yeah, that's really interesting to see how that's shaping, what you're going to do and how informative it was. We're getting a few questions around host genomics and the integration of host genomics. I don't know if either of you want to comment on how you're foreseeing that being kind of getting wrapped into these omics.
I can start maybe just by saying we didn't actually capture permission to get host genomics. So I was certainly interested in incorporating that. I know we can pull it out of the stool samples, but it was kind of a non-starter for us.
Yeah, coming from more of a clinical background, host genomics, depending on the phenomenon that you're actually looking at, it can have a big impact on what's actually happening within the human body. So for instance, people who have metabolic disorders where they can't further process the metabolites that they're absorbing through the gut, it may have some value depending on what health outcome you're looking at. However, again, it's going to be very specific to whatever you're really trying to study.
Great. Just trying to work through. There's quite a few coming through. My apologies. Outside of lyophilization, what other methods could be used to process the samples? Maybe can comment on, actually the downstream processing of these samples.
Yep. So typically, again, if you're using a flash freezing where you're not really adding any solvent to it, lyophilization is still probably the gold standard to dry the samples down. If you are using a stabilization chemistry where you're adding, say, an alcohol to it, simple either evaporator through a vacuum evaporator or nitrogen evaporator, it can actually get quite a bit of the water outside of it, again, so long as it's been homogenized correctly. Even after that, if you really want to dry it down, you can take it and lyophilize it further. One thing I don't recommend trying to do for drying, which can be used in some things, is just direct heat. So again, because so many of the metabolites are either volatile or they're heat sensitive, if you do just try to heat it up, you will lose most of the things and you're going to cause a lot of other kind of downstream effects.
Ken, I'm going to ask this question because I know we're working on it, so you can kind of comment with that in scope. There's someone asking how long the samples would remain stable if they're stored at -80 after being frozen.
This is something that we're looking at here. Our own processes are still in the initial stage. We've only reached the 3-month time period based on our preliminary results. They do seem to be fairly stable after -80 stabilization. And just based on the fact that at -80, there's really not that many chemical effects, and there's definitely not that many biological effects, whether it's happening in a raw stool or whether it's in a solvent. I would be very surprised to see if there's a huge amount of change. Again, so long as the container is correct. It needs to be airtight under the -80 conditions. And again, it can't really allow anything in or out.
Yeah, for the audience, we get that question quite a bit. It seems to be one of these unanswered questions for the metabolomic space about that stability at -80. So we're working on it. And we're almost running out of time, so I'd like to ask maybe a bit of a fun one for you, Jason. Would raw potatoes be more effective than sole milk?
No, I think you'd have to eat a lot of raw potatoes, and I guess when you look across the world, not many cultures have found ways to incorporate raw potatoes into culinary dishes. So I think you need to eat like 3 or 4 raw potatoes to get an effective dose of solnol. Yeah, so I don't know. I think it's easier just to mix a little teaspoon of solnol into your water, your beverage, or here's a plug for our partners. Maybe buy one of the products on our website, and that's probably a more enjoyable way to consume it.
Yeah, actually, I have been noticing, you guys have been showing some success stories of products that are integrating your product into what folks would probably actually see on the shelf at the store. So I don't know if you want to comment on some of where that's actually happening.
Yeah, so the last year has been really exciting for us. Lots of product launches, lots of positive customer feedback via those partners, and I think resistant starch has been kind of known kind of in the background for a long time, and I think we're really starting to see an appreciation for it. It's a very gentle fiber, but it has these real beneficial effects on the microbiome. So just so happens that potato is that #1 natural source of resistant starch. So hopefully that momentum continues.
Great. And I know you guys can see the questions too. And so I just thought I'd open it up if there's kind of a last comment that either of you want to make, around recommendations or thoughts around integrating metabolomics and microbiome together.
Ken, do you want to go first?
Yeah, I mean, it's always going to be a difficult challenge to be able to kind of marry the two omics together on that, again, especially with the costs and the time commitments that are on it. The best I can recommend is try to assess whether you foresee that the metabolites are going to be really important before you start analysis. This way you can at least collect, even if you don't analyze right away.
Yeah, and I would echo that. I think it's important if you want to do especially stool metabolomics. I know that some places, some services will offer certain metabolite analysis in combination with microbiome analysis. And I think based on the initial data we're seeing, that it's really important to, if you want to do metabolomics analysis in stool, you got to collect those samples for that purpose. You can't necessarily collect a sample for one purpose and expect it to work for another.
Yes, I completely agree with that.
Great. Thank you both. I know we're coming right up to time, so that we don't get cut off, I will finish this off by thanking both of you. I think this was really great to see, real world application and really highlight some of the challenges, but I think we can definitely overcome those challenges and lead to some really great metabolomic data that can be integrated into further microbiome studies. So excited to see how much enthusiasm there was. So thank you again to both of you for presenting today. Thank you to our hosts inside Precision Medicine, and thank you to all of our attendees. We hope you enjoyed.